Did you start recording? I can't leave until somebody gets here. Come on, Jack.
Then we'll correct with that.
Hey. Ron already started the recording. Come on. This thing is just...
It's overloaded.
It's just not cooperating today.
Sorry, I have to leave before the end, but they're working.
Well,
That's some stuff out okay if somebody can push the button here.
if you can get him started, I'll be back.
Yeah, okay.
Because my meeting shouldn't last more than
This
an
is
hour.
thing on here already?
Yeah, he's already loaded. It's right here. Chlorpheniramine. So.
You know, he's had trouble sometimes where everything came out. Oh, he got everything right. It came out funny colors and things. But I think that was because he tried to do it on his iPad before somebody.
Yeah, I think he's tried to do it on his
All right.
on his own thing.
Great.
So,
Okay.
and see, this is on, so he can use either one. So everything should be ready to rip when he gets here. And if you'll swap cough over, I'll be back just as soon as I can.
Okay, thank you so much.
Good morning. See, everybody's here.
It's a smaller group this year, but we usually have about 8 people here, but we've waited about 4 or 5 minutes. There's people who come in online, so I monitor the chat. There is chat, which is not terribly common, but so occasionally I'll stand up and ask a question from the back.
Okay.
But I think everything's ready to go.
All right, is there a clock? Someone was.
There is no clock in this room.
That's all right, let's use this.
Yeah.
Okay.
Yeah.
All right, shall we wait a few more minutes?
Yeah, we should wait about 3 or 4 minutes.
Okay.
Water.
I can get something here.
There's, I think, water on the other side.
Collect my honorarium.
Yes, right.
Good morning.
Yeah, we'll be here.
You're from England back in.
Yeah, you're right. You- is that coming today?
Yeah, well, give me a call,
Send you the calls and-
Yeah.
Okay, thanks. Three people wondering for the first 15 minutes. I think there's chat, there's drop off and stuff. So,
How long has this series been running this now? Since, does it start and ending?
it starts on January, finishes in. We actually have just one more session, and then at the end we have people give presentations to each other and we critique them. And then we have a mock study section of that in June. Oh, actually, are you supposed to go to Jean County?
Yeah.
She left the building about 3 minutes ago. All right.
Thanks.
Mhm. Take a look at what's over here.
I've been looking at this for years now, I thought it was pool balls.
Okay, well, we should get started. We have two speakers today. So the first part of the session, the sort of focus of today is tools for personalized medicine type research. And so the first half of the morning will be presented by Dr. Jim Semino, who's the director of the Informatics Institute here at U of A. And the second half will be Bruce Korf, talking about the genomics and related technologies that are useful for personalized medicine. So Dr. Semino is still relatively new on the campus, so I'll just give you a little bit of his background. So he got his medical training in New York City at New York Medical College, did a residency in New York City, and then went and did an informatics fellowship at Mass General Hospital and Harvard Medical School. Joined the faculty at Columbia University Medical School and was there for about 20 years, really developing informatics tools and programs. It's really his passion is figuring out how to make medical records more functional for clinical enterprise, but really more importantly for how to make it so that we can extract information from clinical records in a way that's useful for research. And he, after 20 years in Columbia, he was recruited to the NIH, where he was the head of a division of informatics that developed the information system for the research enterprise in the clinical center at the NIH. And spent a lot of time talking about, he uses terms that I don't know the exact meaning of, so I'm probably not sure this, but really
pay attention.
Yeah, I try.
Pay attention today, maybe, and then you'll know.
Right. So, but he's trying to figure out ways to have coherent terminologies so that people are talking about the same things when they approach the medical record. We recruited him here a few years ago to become the director of the Informatics Institute, and he's the deputy director of the CTS, a member of the executive committee, and has really built tremendous tools for giving people access to the electronic health record here. And as he'll talk about, one of the things the CTSA program in general is trying to support is making it more possible for people to reach into databases away from our local center. Our partner network has several institutions that are close to having accessible clinical databases, but I'm sure he'll talk about what those services are. And part of his focus really is to empower people to be able to do their own preliminary searches at least, and then to know how to get help from the informatics division staff to really take deeper dives into the data. So thank you very much.
Thank you. So last year I spoke at this group on research informatics, and then this year they told me my title was health informatics. So I've expanded the range of what I talk about and necessarily had to cut out some of the details. And to tell you the truth, I'm not sure what is left in and what's left out. So do you feel lucky today, I guess was to quote Dirty Harry. But I'm not sure. We'll see. If I leave anything out, just ask me about it. So health informatics. What is health informatics? It's kind of a funny term. It means a lot of different things to different people, and typically it means something people have a particularly narrow view of health informatics because they've seen it in one context. So even I'm guilty of typically just talking about electronic health records and thinking that's health informatics. Other people will talk about the use of data from health records. Others will talk about bioinformatics. And people, have their own area that they're interested in and often are unaware of or forget that it's a bigger picture. So kind of the blind man and the elephant analogy. So first define informatics, and so this is my definition, the representation of concepts in ways that computers can reason with them. And you won't see this definition typically, but I think when you look at other definitions, this is what they're all really about. We are representing things in a way that computers can manipulate symbolically. Computers don't really reason in the way that we do, but they can execute logic and mathematical functions and that sort of thing. And so if we represent things in ways that match their capabilities, they can help us enhance our capabilities. We have to anticipate how information will be used. We don't try to represent everything in the world. We want to figure out what are we going to do with it and then represent it in a way that's going to make it useful for that purpose. And then we have to prioritize what we represent because we can't do everything. And so we pick the things that are going to be the highest yield. And there's a lot of art to informatics, a lot of art to figuring out how to actually make these things work. I'm going to use culinary informatics as an example, just a general example of an informatics field. So notice we didn't talk anything about health or biomedicine yet. That was just informatics in general. So culinary informatics. So I posed this question to you. Which of these is not a fruit? How many think that watermelon is not a fruit? Show of hands. Watermelon, not a fruit. How many think strawberry, not a fruit? How about almond, not a fruit? Okay, 4, how about tomato, not a fruit? 1, you voted twice. So it was actually only one of, that's okay. It doesn't say pick more than, it's only one. Okay, so the answer is that actually the almond is not a fruit. The others are all fruits. All right, so which of these would you not put in a fruit salad? Okay, watermelon, how many people would well, let's go the other way. How many would put watermelon in a salad? Fruit salad. How many would put cherries in a fruit salad? How many would put almonds in a fruit salad? How about tomatoes? Okay, so knowledge is knowing that a tomato is a fruit, and wisdom is knowing not to put it in a fruit salad. All right. So how do we do that? We do that. You guys all were, the people at home online, there was a unanimous decision not to put tomatoes in a fruit salad. So we all agree on that, whether we agree on what's a fruit or not. We all know that. How does a computer do that, though? If we tell a computer a fruit salad's made up of fruit and here are the 3 fruits, it's going to put tomatoes in. It doesn't know. So what we have to do is a deeper representation of that. So we say watermelon is a berry, okay, and it has a flavor sweet. Watermelons are berries. An arrow, a strawberry, not a berry, an arrow. It's because its seeds are on the outside. So it's called an arrow, and it has flavor sweet. An almond is a seed, also has flavor sweet. A tomato is a berry, has flavor savory. So this is my little definitions, a deeper modeling of these concepts. A berry is a fruit, it has flavors. Arrow is a fruit that has flavor. Sweet is a flavor. Savory is a flavor. And a fruit salad has fruits and seeds and has flavor sweet. Okay, so with this knowledge representation, I could write a little rule that says, helps me pick which things to put in a fruit salad. I won't talk about computer programming or anything like that, but I think hopefully you can see in general, I've represented enough knowledge for a reasonable computer program to be able to decide not to put a tomato in a fruit salad. All right, so that's culinary informatics. It turns out it's a real field to some people. But now let's turn to health informatics. So if we look at informatics overall, and if you Google informatics, 43 million terms, although I'm not sure about this because it was 43 million last year and I'm sure it's gone up. But if you look, culinary informatics, 371 hits, so it's actually out there somewhere. Clinical informatics, several orders of magnitude higher, biomedical informatics, health informatics, medical informatics. These are all kind of overlapping terms. The big winner, though, is bioinformatics. Over half of the instances of the word informatics are associated with bioinformatics. But overall, you can see that the healthcare, health bioscience field kind of owns the term informatics. So when you see informatics, it really came from the healthcare field, and it's sort of still owned in that way. All right, so let's go look at health informatics now. Suppose that I have a piece of information in a record that says the following. The nurse reported that the patient's glucose was 50, which is low, so the physician ordered some glucose and then repeated the glucose. Well, a clinical person would understand reading this that the first glucose refers to a physiologic level in the patient. And the physician then ordered some glucose, a chemical, a treatment, and then repeat the glucose. That's a lab test. So we've got a physiologic parameter, a medication, and a lab test. Yet in the text, it just says glucose. But we understand that because of its context, we know what that means. A computer, though, might not have any idea what to do with that. Okay, so lab reports serum glucose level of 50. How do we represent something like that in a more deeper representation? An order entry, intravenous glucose, order entry, serum glucose test. These are all now, we're representing this thing a little more explicitly. We're using control terms like serum glucose level, intravenous glucose, serum glucose test. And so that's part of it. What if we want to represent that more in depth? So we have a context now where this is appearing. So the lab report is order entry. And then we have control terminologies. And together, that's a deeper representation or modeling of what's going on. And then we could even go further and say glucose is a physiologic parameter, has location bloodstream, has a chemical glucose, and there's a value of a number. Intravenous glucose is a medication, has 2 ingredients, normal saline and glucose, and then it has a strength of 50 milligrams. And serum glucose test is a laboratory test, serum specimen measures glucose. So now we've represented those terms in a deeper way that the system could then figure out something more about these things. For instance, it could pick things from the record and figure out which ones should be graphed because they have a value, and which ones can be graphed together because they're measuring the same chemical, and that kind of thing. So this is now where we're getting into informatics, but not so easy. So what if we could actually write a little rule that says if the physiologic parameter is low, then administer a medication with the associated chemical into the location, and then order a lab test that measures the associated chemical. So we could apply that rule generally to a lot of different things, although in reality we wouldn't just turn a computer loose with rules like that and try to expect it to do the right thing. It would run amok. But what's the location of diabetes mellitus? If we want to model that disease, what's the location of it? Is it bloodstream? Is it the islet cells of the pancreas, insulin receptors, the vascular basement membrane where damage occurs? Is it the glycosylated protein? Do we have a way to mute this?
I do, sorry. It's usually as
Folks at home, if you could mute your computers. We're getting some noise here. Yeah, we don't want to mute me.
Audience view.
You may need to go to attendees.
Yeah.
Are they all muted?
They're all muted.
All right. Thank you.
Sorry.
That's okay. All right. So, sorry, just to recap. So representing things in a deeper level in informatics, not always so easy. If I was trying to represent diabetes mellitus in some way, I'd probably be thinking about how am I going to use that information? So which part of this am I going to actually model? Modeling the whole world, not really practical. So what is biomedical informatics? So the easy answer when you're at a cocktail party and somebody asks you what's biomedical informatics, just say computers and medicine, and they'll nod knowingly and move on. You know, it's hard to explain in depth what we mean by biomedical informatics. This is a definition published in JAMA by Bob Greenis and Ted Shortliff in 1990, and they kind of cram a lot of things into their field that concerns itself with cognitive, information processing, communication tasks of medical practice, education, and research, including information science and technology to support these tasks. So pretty broad. They cram a lot of concepts in there like cognitive and communication that maybe you wouldn't have thought of right off the bat, but there's a real important aspects, and there's probably even more that they just didn't cram in there. This is mine, the art and science of organizing knowledge of human health and disease and making it useful for problem solving. So that's the end task, and problem solving is very general. Are you trying to cure a disease in a patient? Are you trying to generally find a cure for a disease? Are you trying to figure out how to sequence a genome? Those are all problems that we would want to solve in this process of ultimately improving human health. Okay, so I have this diagram. I don't know if you can make it out. We start with the sort of biochemical level. We've got genetics and molecular biology, and the term bioinformatics is often used for that level. All right, so when you hear bioinformatics, sometimes it means that. Sometimes it's just a term people use for anything that has to do with computers and medicine because it's an easy term to say. And in fact, for a long time, the NIH would say bioinformatics when they meant all of biomedical informatics. And I was there and I kept saying, so is this the National Institutes of Biology? And they eventually stopped saying bioinformatics. So then we've got, where we move up to the sort of cellular and organism level, we have translational informatics that takes what we are doing at this level in research and moving it up to research at a higher level. When we get to an organism, like a human, and we're looking at physiology, pathophysiology, we talk about clinical research informatics. So the informatics around performing clinical trials. So that could be something like just a computer system that captures the data, or it could be an expert system that tells you what to do next in a complex protocol.
So would medical claims data fall in that?
So no, because we're still at the research level, but now we get up to patient care, and then your medical claims data would be at that level, and that would be clinical informatics. And the term medical informatics was often sort of synonymous with clinical informatics, but then they tried to make it broader, and then the biologists said, well, we don't do medicine, so medical informatics doesn't apply to us. And so the biomedical informatics or health informatics became a more predominant term. And then there's one more piece, which is population informatics. So we get up to the public health or population health level. So there are informatics at all these levels, and things that apply in one area can be often used at another level. All right, so there's a spectrum of problem solving. We start with the basis, science base in biology and medicine. We do collection of data interpretation of signals. We take data, turn it into, people will talk about turning data into information and information into knowledge. You know, that's a semantic game that you can play. I think information subsumes data and knowledge, and other people think it's a sequence. But the important thing is that you're taking raw things and turning it into something more symbolic that you can then think about conceptually. Application of science and data in clinical care, so now moving up to that level and ultimately out to population. So it's really the spectrum of problem solving. So we solve something in a laboratory that says, oh look, this drug causes this gene to, to produce, ultimately produce more of a protein which helps treat this disease. So now we can say, let's give that to a patient, or let's put it in water and give it to everybody. And so we take that information and move it up the chain. So I have a case presentation that has a lot of stuff in it that can be relevant, I think, to a lot of different levels of informatics. And I guess you guys will get the slides, right, and provide these slides so you can read this at your leisure. It's a really compressed case, but it's got a lot of interesting things in it, I think. A 50-year-old Native American female presents the emergency room with chief complaint of lip numbness, nausea, and chest pain. Well until half an hour prior to arrival in the emergency room, eating at a dinner at a seafood restaurant in Rock Harbor, Mass. She was finishing the dinner of New England clam chowder, lobster, steamed clams, and corn on the cob when she noted onset of symptoms. Others in her party ate fish and chips, although 2 other people ate the clam chowder. None ate the steamers. Gives a history of hypertension. She takes this half blue, half green pill. She was treated in the past with tuberculosis when she was pregnant. Doesn't remember what she was treated with. And she was seen at another hospital where she had a liver biopsy. Thinks she has hemochromatosis, and she's allergic to buffering. All right, so we've got presenting complaint, history of present illness, past medical history, rolled into this little page. Then we start collecting data about the patient. Physical examination, well-developed, well-nourished, diaphoretic, meaning sweaty, female, moderate respiratory distress, and so on. We have vital signs and other physical findings, and then chemistry results. And so we have glucose of 100, another glucose, plasma glucose of 150. We've got complete blood count. We've got a finger stick blood sugar. So we've got blood sugars represented in a number of different ways here as a serum, glucose, and finger stick result. And then there's a urinalysis, also has a glucose in it. And then a blood culture positive for methicillin-resistant Staph aureus, and a cardiogram showing sinus rhythm with a rapid rate, axis minus 30 degrees, ST segment 2 millimeters, and so on. So this is, now we've got some data that we've collected on the patient. Some of it is, so this is objective data as opposed to the subjective data that the patient provides us. And then finally we have the hospital course. Nurse reports the patient is more worried about who's going to care for her father if anything happens to her. Medical student reviewing the case wonders whether paralytic shellfish poisoning, which is what this patient had, could cause a myocardial infarction, and decides to do a literature search. The patient is treated with activated charcoal and stomach lavage, followed by enteric-coated aspirin because she's having a myocardial infarction. Due to worsening respiratory insufficiency, intubated and placed on mechanical ventilation. Well, she was allergic to bufferin, and they gave her enteric-coated aspirin, so maybe that wasn't such a good idea. And so that's the case. So if we look at the data types in there, we've got things that we collect from the patient. Some of it's in text form, some of it's more objective and quantitative. And then we also have signals that we collect, and we usually take the signals and turn them into some coded representation like text. So images and EKGs, we turn that into text. Then we start coding that information using control terminologies, either with manual methods, or we can use natural language processing to produce coded data. And if we're lucky, we can get it all the way to standard, using standard coded data, which allows us to pool data across different sources and share it, like claims data, for instance. So these are blobs to the computer. They're just things the computer stores. You say, hey, give me the image, give me that text report. The computer goes, here you go. It doesn't really know much about it other than it's supposed to store it and who it belongs to and who's allowed to look at it. But these things are the symbols. And this is where you get to the point where the computer can start to be helpful. So now in informatics, we would try to answer a bunch of these questions. So how do we find out what medication the patient's on? We have this weird description of a half blue, half green tablet. How do we get our records from another institution? And how do we get the right records? Make sure it's the right patient. We don't have universal patient identifiers. So they don't seem like clinical, health challenges, but they are challenges nonetheless to the practice of good patient care. How do we keep the clinicians from getting overwhelmed by all the data that we provide? How do we recognize our potential allergy to bufferin or to aspirin? And how do we get relevant evidence from the medical literature? So we have a medical student doing a search. How can we help use data from the record to help us get that relevant information? How do we take expert systems and apply them? So maybe we can use rule-based care to tell us things like, hey, don't give that patient aspirin because she's allergic to bufferin. How do we assess her genetic predisposition? So eventually we're going to have the whole genome as part of the record. How are we going to make use of that information to help us decide, what was this patient's predisposition, what's the right medication to use, does this patient have hereditary hemochromatosis, and so on. What do we have to report to public health and why? And how do we report it? So this is paralytic shellfish poisoning. Are you supposed to report that to the health department? And if so, how do you transfer information that way so that then it can be applied to the care of the public? And then how do we take all this data and get new knowledge from it so the next time somebody comes in, we can go, aha, this patient looks like that patient that came in 10 years ago with chest pain and lip numbness, and it turned out she had paralytic shellfish poisoning and got a myocardial infarction as a result of that, and it could use that information to help us figure out, well, what worked for her, what didn't work, and let's apply that to our current patient. So those are all questions for informatics. I'm not going to try to answer them now, but I'll try to help you understand some of the pieces to the puzzle. So we might be interested then in reusing data from electronic health record for a variety of purposes, whether it's the immediate care of the patient through something like a decision support system or expert system, or for a learning health system where we're going to try to understand more general principles of health. So why do we use this a lot? And David alluded to this as well in providing data to researchers who are interested in either looking for patients that meet certain enrollment criteria or understanding the natural history of disease. People like to use electronic health record data. They often use claims data for this purpose. And so what's the big attraction? Well, first of all, the data are collected by clinicians and instruments, so they're generally high quality data. They're, the instruments in particular are usually very highly standardized and regulated, calibrated, so they produce reliable information. And clinicians, for the most part, are trained to be observant and collect data in a way that other people will understand. So when I describe a heart murmur as a grade 4/6, anybody else who knows about heart murmurs will know, oh, that's a loud murmur, and it has a thrill. You can feel it, but it's not so loud that I can hear it without the stethoscope on the chest and so on. It contains data about the phenome, so we're very good now at collecting genetic data. We take a little sample, we send it off somewhere, we get back, terabytes of data about the patient, and from that we figure out their genome. But then what does that mean in terms of what's really going on with the patient? For that we need the phenome. We need to know how it's being expressed in the organism. And so the EHR is a good place to get those kinds of data. They're becoming universally available thanks to the Affordable Care Act. There's been a lot of promotion of adoption of electronic health records, and so I think something like 85% of hospitals now in the country have electronic health records. So more and more of those data are at least captured and somewhat available, and it's almost free. So it's already there, it's already been captured, it's an electronic form. All you have to do is pay some programmer to write a program to extract the data from whatever the database is, and then that program can keep running, collecting more data for free. And so really the cost per byte of data is going down dramatically, and the bigger the data set, the lower the cost per element. We can use it for things like post-market surveillance or phase 4 studies. So if I'm not familiar with this, a drug goes through the approval process, the FDA, and then it gets released. And so now it's out there, people are prescribing it, and a phase 4 study would be looking for complications or adverse effects associated with a drug that weren't discovered during the initial studies because they only studied 300 or 1,000 people, and this thing occurs 1 in 10,000. Well, now you're giving it to millions of patients, and you can discover, for instance, that people who take this drug are more likely to get myocardial infarction or ruptured Achilles tendon or other weird things that you wouldn't have expected from the way the drug is supposed to be working. You can also use it to validate research studies. So if you have a research study that says, oh, patients who get drug X do better than patients who get drug Y, you can then go back and collect data and say, let me find all the people that are getting X and Y and see if that's really happening in the population. Now, it's a retrospective study, but you might look at that and say, yes, we're seeing what we would expect, or no, we're not seeing it, and then you can say, why are we not seeing it? Is there something different about these populations, or what have you, and allow you to question some of the outcomes of prior studies. You can also use it to replicate research studies in what are called pragmatic trials, where you say, there's two drugs out there. Doctors are using one drug or the other, and mostly they decide based on what it says on the free pen they got from the drug company or who bought the pizza for the CME conference. And so it's really arbitrary how they assign these. So why don't we go to those doctors and say, listen, you're about to treat this patient for this condition and there's two choices. Why don't you let us choose randomly? We'll assign it, and then you just give the, then you write prescription and do whatever you want. And the physicians will say, yeah, I don't really care whether I give this drug or that drug, fine. So now you've got patients randomly assigned to this drug, and you can do this at a large scale. You can do it automatically. Just have an alert that pops up when they order a drug and say, hey doc, your patient's eligible for this study. Would you mind doing this and consenting the patient and so on? And now, instead of paying $10,000 per subject, you know, it's costing you $100 per subject, and then you just turn them loose. You have an intention to treat study, you let them go, and you just pull the data almost free, and you figure out what's going on with patients. So that's a pragmatic trial. Very appealing for studying things, that for, especially for drugs that are already approved where there's not a big market for us to try and go back and look at them. But it's important to understand the limitations of electronic health record data. So that's part of where informatics comes into. So when you're using claims data, for instance, is that good enough for, you know, for your study, or do you need something else? So understanding those limitations is an issue. I want to talk about now about I2B2. So I did leave this in. And David mentioned providing tools here at UAB for access to patient record. So I2B2 is one of those tools. So we have an electronic health record here. Some of you are wearing white coats, so I'm sure familiar with Cerner Impact. And so that's our electronic health record. It has a big database. Then we extract data from that and put it into another database called the Enterprise Data Warehouse. And that Enterprise Data Warehouse can answer questions either for operations of the hospital, for quality assurance, or for research. And we have EDW, Enterprise Data Warehouse analysts, who are trained to go in and pull data out of those records. And you as a researcher can get that training, but it's pretty tough, and you're better off working with the professionals. And so a researcher will typically interact with an EDW analyst to say, find me all the patients that have, you know, these particular characteristics because I'd like to recruit them for a study, or I'd like to see how they're doing on which, you know, various drugs, or see if they're getting some kind of adverse events that I would be looking for. So we can do that. Now, it's a little tedious to do this because you go to the IRB and you file a, you know, you want an exemption because you're just getting limited data sets or you're getting summary data, and then you go to the analyst and you say what you want. The analyst comes back a couple weeks later and says, okay, I finally got to your thing. Here's some data. And you go, that's not what I wanted. And you go, well, that's what you said. And you said, well, this is what I really need. Okay, I'm sorry, let me go get it. And you work back for it. Maybe you have to say, oh, and they say, oh, you didn't tell the IRB you wanted these data back. To the IRB, right, and doing all this stuff. Eventually, hopefully, you get the data that you want. It takes time, hopefully high quality data. But there's a faster way, and that's to use something called I2B2, integrating informatics for biology to the bedside. And it's a lot of different things in I2B2, but the one we're interested most right now is its data repository. So we extract data from the Enterprise Data Warehouse, put it into I2B2, and then researchers can interact with it directly. And I can talk more about that if you're interested. But then you can get out data that are about our patients, and then we also are setting up a network through something called Shrine that will let us get to other data in the Southeast. Shrine network, which is hospitals throughout the Southeast, and then we're actually joining the Accrual to Clinical Trials network, which will give us access to data in something like 60 or 70 hospitals.
When is going to happen?
When is what can happen?
The connection to Shrine and other.
So we're waiting. It's, you know, soon. We were getting there. We had the agreements in process, and every time you add another institution, some lawyer changes the thing, then you got to go back and get everybody's approval, and then everybody has to sign it, and it was driving me crazy. So I kind of put it on hold, said, look, we just joined ACT. They have their act together, pardon the pun, but they've already done this, so they have an agreement that we can sign on to, and I'm just going to go with that, and then you'll be able to go to the whole network, or the members of the Shrine network are all, our Shrine network are already either in ACT or joining ACT, so you'll be able to get to those data as well. And the reason you might be interested in the Southeast one is because if you're trying to recruit patients that are close by for a multi-site study, or that could actually, you know, travel, you know, a 6-hour drive, you know, if it was worth it, you'd be able to do that. So you might be interested in a regional network, or otherwise it's national. But, you know, I'm hoping this ACT thing we're gonna get this in the next few months. We have a kickoff meeting next week, I think, to talk about the technical aspects. we already know how to do the technical aspects. We've already done them here. We've already installed the act ontology. We've made it work with the other Shrine nodes, although they haven't made their nodes work. So we're pretty confident we'll technically be up and running. Then it's just a matter of getting everybody to sign off on that. And that'll take a couple months probably. So hopefully by the end of the summer. I didn't say which summer. Okay, so what can you use I2B2 for? Well, there's a lot of different pieces to this puzzle, and it's kind of a cycle. You can start with exploring a hypothesis. You can you can do feasibility assessments just in terms of looking to see if you have the number of patients that you need for enrollment. You can you write your proposal. You decide whether you want other institutions in it. You can start with limited data sets and recruitment data sets. Then eventually you have to get IRB approval and get a data set that's going to be used for your research, and any additional data you want to collect. So each of these systems contributes to that in some way. So the I2B2 and Enterprise Data Warehouse collects data that are retrospective, data that have already been collected, so they can be used for these things. And the other institutions is where you start to look at Shrine. And so when you just want retrospective data, these are the I2B2. If it's in I2B2, is probably ideal because you have it under your own control. When you start collecting data prospectively, then you may need to be using the Enterprise Data Warehouse if it's more complex, or even going to Impact. If you, for instance, say, well, everybody that comes into the hospital I'm going to write orders in the order entry system, and the lab's going to do its thing, and all those data are going to go into their record in Impact. So you may need to get the data directly from there. That's just opportunities for outside research. So it's kind of just think about this cycle of how we go through, and we go through all that, and then we come up with new hypotheses, and we start all over again. So it's an I2B2. This is hard to read, but it's you'll have the slides, and it's the big ones are things like lab orders, almost 142 million rows of data, blood pressure measurements, 48 million, home discharge medications, 11 million. Counter insurance data, 11 million, and so on. And then we have calendar years. It goes back to 2010 for the most part. We have some older data that come from other sources, but most of these data are from 2010 when we installed the Cerner system. And every year we go, we get more data. So when I got here in 2015, 2010 wasn't that long ago. Now it's 2017. It's 2 years more of data. So it's really building up. And of course, those older data become less useful as time goes by. Some of them may be useful, but the real values usually goes up with the recency of the data.
So are they pulling that based on like CPT codes, or how are they?
No, this is all the data on all the patients in the system. So we have CPT codes, we have ICD-9 codes, we have lab data, all of those things, but it's on all the patients. Now, if you want data on just, who asked the question? I'm sorry, I didn't see. Yeah, so if you wanted data just on people with particular CPT code, you could say, find me all the people that have the CPT code, and I'll say, okay, there's 11,000 of them. You go, okay, now get me all their lab data, or that kind of question, so that you can create your own filters based on those codes.
So then, so the reason that I'm asking, if you want to talk afterwards, that's fine. I'm interested in codes that reflect palliative care, and there's not a standard as yet. So information is being entered differently, sometimes under hospice, sometimes it's by a procedure that you don't know if it's for palliative care or something else. So I'm just wondering if this would capture that.
So when I mentioned the caveats about using electronic health record data, one of the caveats is to understand where the data come from, right? So it's not magic. The EHR doesn't magically know everything about the patients. It's not Google. Where it knows everything about you. It knows what it knows. So the first question to ask is, where are those data captured, if at all? If they're not captured anywhere, it ain't going to happen, although you could find some things that might make you suspect that somebody's getting palliative care. But the reality is, I think, in the electronic record, and I don't know specifics here how those things are captured, sometimes there's an order that's written that says patients palliative care, or comfort measures only, or something like that. There might be an order written. Now that order might be text. It might just say other, and then there's somebody's typed in, comfort measures only. So now you say, aha, I have to look for other orders, and I have to look for this. And it may be represented in multiple different ways, in the same patient or in the same system. So for instance, it might be in a discharge summary. It might be in an admission note in the text. It might be in the problem list or the assessment list. It could be in a nurse's note. So it could be in a lot of different places. all of those data are accessible. Not all of them are accessible through I2B2 right now. I2B2 has this set of data, and there's a lot more, and we're working on that. The other thing we're doing is natural language processing to extract features from this. So for instance, if we have a concept called palliative care, we would process notes and we'd be able to figure out which notes either say that explicitly, and that's just an index, but which ones mention it. So there could be, synonyms of palliative care like comfort measures only, or whatever other synonyms there were, other phrases, or that might be in there. We would boil those down to the concepts that are in the text. So applying natural language processing will give us a better way to get at that. We're looking at expanding the control terminology that we have for representing the record, so that even when stuff is not captured with controlled terminology, like in a problem list where people pick specific things, we can say, okay, this patient doesn't have it in her problem list, but she's got it in her record, and we'll be able to say, find me all the patients with this concept somewhere in the record. Then you would probably just manually review the record, but it would at least boil it down to a smaller number. So we're getting there. I have good job security. It's going to take a while. All right, so let me show you I2B2 a little bit, and I don't see what time it is. Is it? Okay. I'll just talk faster. So let me show you a little bit of I2B2. So this is the I2B2 screen, and on the left-hand side is the term lookup application, where you pick the concepts that you're interested in, and then you can either navigate through a hierarchical terminology, or you can type stuff in and it'll pull it up and match things on based on name. So here I clicked on if I had the navigate terms, which is that leftmost tab at the top, it would show me a whole tree structure that I could navigate down. Or here I've clicked on the find tab, and then I've said search by name, and I've said containing proton pump, and I've said I want medications. So I put in the word proton pump, and I got this class, proton pump inhibitors. And then underneath is seven drugs names, and there's a number next to each of those, and those numbers are the number of patients that have that somewhere in their record, in their medication list. So I can drag those over and build up my query. Now let me go over here and let's see. Folks at home can see the mouse, I hope. Can you guys see that too? So before I did the proton pump inhibitors, I must have typed in gastroesophageal reflux, and I found a term, and I dragged it over here, and now I put that in there, and it says there's 37,077 patients with that term. This is ICD-10 code K21. And then I got my proton pump inhibitors, and I started dragging, and I dragged 4 things over here into this thing, and there's an and here. So when I run my query, it says there are 25,776 patients. So there's a subset of these patients, 37,000 of those gastroesophageal reflux disease patients. 25,000 of them have one of these 4 proton pump inhibitors. Now, I could also do it this way. So instead of dragging over the individual things, I can just drag over the whole class. And now I've got 30,000 people out of 37,000. So I picked up another 5,000 people that are on proton pump inhibitors of some sort and have gastroesophageal reflux disease. Now, that gives me a number. That's a summary data, and maybe that's all I want to know. Like, what percentage of patients at UAB, whoever had this diagnosis, have ever been given a proton pump inhibitor? It's an interesting to do the query. What percentage of patients who are on insulin have the diagnosis of diabetes? And it's actually not what you'd expect. So it's not clear why that's happening exactly, but it's kind of universal. A lot of places have reported this kind of data. But the summary number might be useful, and then you can get demographic breakdowns of this. Let's see if I have it. I don't have a picture. This is just a little bit more about the query. So I can say, if I'm picking a drug, I can say which dose of the drug I'm interested in. Here I was picking, this is I think the body mass index, and I can say I want it to be less than a certain value or greater than a certain value. Or here's a laboratory result. I could say I want it greater than or less than or between a certain range of values. Excuse me. I can say I'm interested in tests that have certain units of measure, that kind of thing. So I can limit which things I'm interested in a number of different ways. And then here's another aggregate study, and this one is, let's see, sickle cell anemia, sickle cell disease, not with crisis, and sickle cell disease without crisis, a bunch of different terms. And because ICD-9, ICD-10 have slightly different terms for these things sometimes. So I said, basically get me a bunch of sickle cell patients and that have a creatinine greater than 1.5, or 1.5, and I found 107 patients. Okay, so I've got my summary, and then I can get more details about the patient. So it's giving me accounts, how many are male, how many female, how many are alive or deceased, and so on, and I can even get it, graphed out in various ways by gender and race and ethnicity, that sort of thing. Now, that's nice to have the summary data, but what if you want detailed data? So normally, you go to the IRB and you say, I'd really like this detailed data on patients with sickle cell disease and creatinine greater than 1.5, because I'd like to see how their creatinines have changed over time, let's say. So, and they'll say, well, do you need to know who the patients are? Do you need identifying information? You go, no, I don't need identifiers. They go, well, then you could just do de-identified data, and then you don't need IRB approval. That's not human subjects data if it's de-identified. And you say, oh, okay. And then they go, wait. Didn't you say you wanted to see how it does over time? So you need dates. So how are you going to handle dates? And you say, well, just look at the dates. Those aren't identifiers. And you go, well, they're considered useful for re-identifying patients. And so when you have dates, that's called a limited data set. And you go, well, now what do I do? And the IRB goes, no problem. You can have a limited data set. You don't need to get consent. We give you an exemption for the limited data set, and you have to do you have to agree to a data use agreement and do all these things. And so you fill out a form and you check off some stuff and you tell them about your study, and then you fax it to them or whatever, and they approve it and they send it back to you, and that takes a week if you're lucky. All right, and then you go in and you get your data, and then you find out, oh, I need different data, and you have to go back and around. Well, what we've done is we streamlined the process with I2B2, because what we say is if you agree to the data use agreement up front, then every time you want data, just tell the IRB what you want, and they give you automatic approval, basically, because you've answered the questions in the right way. And so then you can get your detailed data that way. So when you go in once you've done your summary, you fill out this little form, and basically you just say what kind of data you want. You put in a title of your study. You put in a little description of the study, what it is that you're looking at. You also say who else you're going to give the data to. And if you as a researcher have already done the human subjects training and privacy training and also signed the data use agreement in the learning management system, and then so you're allowed to get the data. And anybody else who's done that, you can put in their Blazer ID and say these people will also get it. And the system will check and say, no, this person hasn't done the training. You can't give it to them. But if people who've done it, then that's fine. And now you can get your data, and then you can say what kind of data you want. And you get your data as a spreadsheet that looks something like this. And we're changing this, we're updating this all the time to try to make it easier to define what it is you want to download. But basic idea is the same. You'll get a tab delimited file that has an identifier, a patient identifier, but it's a temporary ID that's only useful for that data set. So if you have one file with drugs, another one with labs, another one with admission diagnoses, and that kind of thing, it will be the same identifier across those tables. But next time you do the query, the identifier will be completely different. And then you can get labs and actual dates and actual results. And then you can do your study. This is just a different format. There's another one. This is showing diagnoses, and it's got the code and the name and so on. This is let's see, do I have time to do this? Yeah, so this is just a little demo. So let me run this. It's going to let me do it. Somewhere in here is a play button. I thought. Not going to work. Let's see. Okay. Well, there was a video, but it's not cooperating.
There it is.
Oh, is it running?
Up at the top.
Oh.
Click at the top.
Oh, here, this? Oh, okay. Oh, yeah, I've got a link out to,
Maybe control.
let's see what it's going to do. That will just take me to, that's if I want to run it myself.
Oh.
I don't want to do that. I think I've got another link to a YouTube. There it is. Let's try the YouTube version. Let's see if that's going to work. I hope folks at home can see this. If not, you can just go to UAB I2B2 and you can find a demo of this. So I'm typing my name and my password. This is my Blazer ID and password. And I go in and I have to pick the I2B2 host that I'm interested in. There's only one, so you just say select. And now here's that that browser. And at the top, you can see there's the navigate terms. There's some other things that I hid in my slides, but now I'm going to pick diagnosis, and I'm going to type in systemic lupus. And I say find, and it pulls up a bunch of stuff. And if I scroll down, patient, eventually I will find the term I'm interested in. And there is systemic lupus erythematosus. So I drag that over, and it shows me there's 3,183 patients that have that diagnosis. And now I'm interested in lab tests. So I'm going to type in creatinine. Click find, and it pulls up a bunch of different lab tests that have the word creatinine in it. And there's one right at the top, says creatinine blood. And I pull that over. Oops, come on. And then I can set the range, and so I want it to be greater than 2 in this case. And now you see it's and over here. And so I'm anding those things together, and then I say run the query, and I say I want different kinds of data. Check off those boxes for race and vital status and so on. I run my query. It's running. I edited the video a little bit. Let's see, 6 seconds. Suddenly it's 34 seconds, 36 seconds, and then we got our summary data. I don't know how I missed the summary data somewhere. And then I go to download the data, and I can get let's see, this is the demographics, and I can say view result. And there's a query again, and then I get these nice graphs. They're not showing up too well here, but there's age buckets and gender, and let's see, race and language, and so on. Marital status, vital status, how many are alive? 349 people alive. So if I was interested in recruiting people with renal insufficiency and lupus, I'd have 349 people I could look up. And now I want to do the detailed download, and I get this screen, and I fill out, I want patient counts. I can select code family. I'm going to say I want in lab orders, or pick lab panel, sorry, and go down, and now it's already typed in. I said renal insufficiency in sickle cell, systemic lupus, and it says natural history study of people with, and I'm going to type in that, and it doesn't have those pre-filled in. That was left over from the last time I did it, and so just fill that in a little bit. I can put in other people's Blazor IDs, and then I say which of the queries that I've done do I want the data from. I hit submit, and that's the end of the video, because the next step would be to see the actual files. Okay. Any questions about that?
And so with that new sheet there for the IRB, it's automatic, or it takes like...
It's automatic. No. You do it. It's just like what we saw. It's real time. And then if you decide you wanted to get something different, you go back in and you do another download. And the form's already filled out. As you saw, mine was still filled out from the last time. And so you don't have to fill it out again if it's already there, and you can just keep doing that. These don't actually go to the IRB in any physical way where they review them and sign them. It's documented, though, so that if they wanted to show that they were overseeing what you're doing, they have that documentation where you've agreed to the various things.
Maybe you could mention just briefly what people have to do to get trained to be able to do this.
So to get trained to do this, you have to go to the learning management system. You have to take all the normal human subjects training that you would have to do. And you have to do one more course in the learning management system. And if you just go in there and type in I2B2, you'll find the course. And the course is basically read this document and then take an exam. And the exam is one question. Do you agree with this document? And the answer is yes. Okay, and you click yes, and then you are certified that you've read this agreement and you promise to abide by it. The agreement basically says things like, I promise not to give these data to anybody who's not authorized to see them. I promise not to try to re-identify the data or to try to contact these patients or to continue to repeat this query to get more data. I'm only using data that's what's called on the shelf, which is what the IRB allows. So you're agreeing to, and they're not owners, and that's why typically when you fill out the form for the IRB, you're always saying the same thing. So we just said, let's let people do it up front.
Is there anyone who can help if you have specific queries? And
Yeah, we have. So if things aren't working, you can contact the CCTS data analysts, and they will help you do this. And you may have to do that anyway if the data that you want aren't in I2B2 yet. If you wanted notes or you wanted some data, we're continuing to add new data to I2B2, but you might find you need something more detailed than what I2B2 will give you. But I2B2 will at least allow you to find counts of patients that you can then go when you're writing your proposal, say, well, we've documented that there are 343 people here with lupus who are alive and have creatinine greater than 2, and we're going to try to recruit 10 of those. And that, that enhances the credibility of your study, because there's a lot of sensitivity around investigators who create these grandiose plans to recruit patients, and then it turns out they don't have anybody with that actual condition at their institution, or they don't have enough to recruit. So some caveats and let me see, I'm running out of time here. Let me just see how many slides I've got left. 8 slides, so that's just 4 minutes.
Can I just ask a question
Yes.
related to what you just said?
Mm-hmm.
So if there are data that's missing or limited, can you match this data set with other data sets?
If you want data, so if you do it through with the analyst, then you'll probably have to, for now, you'll have to get IRB approval, because now they'll be looking at identifiable data. But you would almost certainly get it with a waiver of consent, because the analysts will act as honest brokers, and they'll remove you don't care about if you don't care about the identifiers, but of course they're needed to match it up with other data, then they'll see the identifiers, but they'll remove them from the data set that they give you. So they act as honest brokers. If you wanted to do things like contact the patient or match it up with data that you've collected from your own research where you have identifiers, then you have to go to the IRB, and they'll have to make a determination of whether to give you a waiver of consent for your new study. If you're only using previously identified data, and of course they're already your research subjects, they're going to probably say yes. If you're generating new data, for instance, getting specimens that we've saved in the biobank, and generating new data, then that would be different. And of course, if you want to contact the patients, you have to identify them, and then that would be part of a new IRB-approved study that included a recruitment process. And you would explain to them, well, we're going to go to I2B2, then we're going to go here, then we're going to get the data from, we're going to get the contact information from the warehouse, then we're going to call them up, and so on, and that would be part of your standard IRB process. So caveats, I mentioned there's some caveats. Electronic health records can have incorrect data or inaccurate data. So a patient may tell you they have an allergy to a drug, and we put that dutifully in their allergy list, but in fact they don't have that allergy. They often don't tell the complete story, because patients go from institution to institution, and although we do have a health information network in our health information exchange in Alabama, it's very poorly subscribed to. So it's really difficult to get all the electronic data that's been collected on a patient. The data may have been transformed, so for instance, if they're turned into billing data or claims data, they're often not useful for clinical research and trying to understand things at the level of granularity of what's really going on with the patient. Data, as I mentioned, data captured in text notes may be difficult to get at, and the EHRs may have data from multiple sources, and you have to figure out, well, what does that mean? Are these data comparable? So if some patient if we have an outside lab, we have an inside lab, we have a patient telling us some lab results, we have a handheld machine, somebody transcribes the data into the system, there may be some effect on the ability to compare those data with each other. Data granularity may not match the needs that you have in your research. A typical example is pain. So I see a patient, hey, how's your pain? And she says, oh, it's much better today. Well, I write in the note, the pain's much better. But the researcher might say, no, I needed a scale of 1 to 20, and where 20 means this and 17 means this and so on, and it has to be captured at a different level of granularity, so you may not be able to get that from your EHR. And then the way that protocols are carried out is often inherently different than traditional patient care, because in fact we're trying to find better ways to care for patients, so we may break the rules, or we may collect data differently. So we may collect data every day on somebody in a study, whereas normally we would only do it once a month or maybe once a year, and so the kind of data you get in an EHR may just not match up with the kind of data you would expect to have in a research study. All right, so concepts of health informatics, it's about representing data and knowledge, it's about controlled terminologies and other standards. I mentioned the signal processing, the way we take the raw data and turn it into concepts and terms. And I mentioned natural language processing. There's also mathematical modeling, decision analysis, lots of other tools that we bring to bear in health informatics. Even user interface design and then building of knowledge sources that can be applied for decision support. Think of PubMed. All right, familiar with PubMed? So that's an informatics tool, came out of the National Library of Medicine. And they've been collecting data on publications since the 1850s. And then they computerized it back in the 1960s and then finally put it up on the web in 1994, 95 time frame. So those are all informatics tools. Application areas can go from biomolecular modeling and image processing to electronic health records. We didn't talk about telemedicine, public health information systems, consumer informatics, disaster informatics, research informatics, which is the I2B2 program, education informatics. So all of those are different areas within health informatics. So I'm going to leave this for you guys to look at, how all these things interact. We spent too much time on I2B2. Or maybe we spent the right amount of time. But you can look at all these different concepts and how they integrate with each other or interact with each other. And and again, forces compelling. I'm sorry I'm out of time. Yep, I don't want to cut into Bruce's time. But lots of different things that hopefully you read about in the newspaper. I certainly see them in the newspaper, but of course I'm focused on these kinds of things. But these are things that you'll typically find are becoming social issues that relate to the use of health and information technology in healthcare. Crammed way too much in here. Okay, if you want to learn more, American Medical Informatics Association is a good place to look. Here's their website, amia.org, and they have a conference in Washington this year, November 4th through 8th, and it's a 3-ring circus. You can go there, and everybody doing anything in informatics will be at this meeting, and you'll see what they're up to. And so if we go back to our blind man and the elephant, it's really, there's lots of pieces to this elephant. It's not just information, it's not just data, it's skills, it's the social issues, all these things fit together to make it all. Okay, that's it. Maybe a minute for questions, but then I got to turn it over to Bruce.
Can you look up imaging reports or specific phrases from imaging reports right now?
Specific, you can't, not yet. That's one of the things we'll be doing. You can't, but you can request all the imaging reports as a data set and then search on those yourself. Or you can ask the analyst to do it. Say, find me all the chest X-rays that have nodule in them, or something like that, and they can get that. And get all the diagnoses that patient has, right? Whatever it is, hundreds of thousands or millions of patients.
So if people need help, they contact Informatics Institute?
No, no. If you need help with research informatics, you would contact CCTS. And CCTS, the Research Commons, and there's an informatics link there. And the informatics staff can help you. The Informatics Institute is a collection of researchers, many of whom are related to the CCTS, but we don't provide the service. We're teaching some courses, working on developing degree programs. We're doing informatics research as opposed to biomedical research. So you might end up contacting us if you were interested in a postdoctoral fellowship or changing the theme of your PhD program. But if you're looking for research data, then you go to CCTS. Okay, thank you.
Thanks. This
Yep.
is very great. So we'll take a 2-minute break. We need to start pretty soon, so take a moment. Thanks very much.
Okay. Yeah.
People otherwise never ask questions, but you can see everybody generally have moved on to
So what kind of
decision. I think I have to let go soon, I'm sorry, but Gene will come.
So we're not
Good,
doing
but you can't use the mouse to grab this.
Right.
Oh, yes. I mean, I do. Yeah, so it's yeah,
CCTS provides, analysts should be able to work with this practice. Start. You can also
really schedule it every hour. Yeah.
There should be a link on CTSTP.
Standard track to show the time.
Is a way for researchers center. And say, I have this problem, I need informatics help, and what's the best way to do this? It could be questions about the collection methods, or
Stand you order first.
the researchers who come to those who say, oh, I'll collaborate with you, I have to, but at least that's what it is, when you send people to do that. So you can contact Jennifer Croker, she's a PR. Okay, so CROKPR, or you can just go to the Research Commons page at CCTS. I know there's a level link there. To contact them, to say you're interested in research. Gateway.
Yeah, you just email CCTF at UAC.edu. She'll end up getting it probably to. Sorry.
You did.
Sorry.
Okay, I sent him a list of a bunch of patients. He should have,
I think that's really just what I
And then we had, the way that I read my database was based on courses and radiation. Some patients had more than one course, but there was about 14,000 courses.
So I don't know the official names of some of the things that we're in charge of, but I'll just introduce really briefly and we can correct anything that's egregiously wrong.
Titles are
Sure.
unfortunately, the way that the is that it's one that's non-small cell looking. But it's a radiosurgery, it's rarely do radiosurgery for small cell. So you can only like the recurrent. So there's probably there's small cell.
Okay, we need to get started, please. Okay, so the second half of today is to talk more about kinds of research that lead to advances in personalized medicine. The speaker is Dr. Bruce Cork, who's the chairman of the Department of Genetics here at UAB. He's a pediatric neurologist who's got personal strong research interests in neurofibromatosis and has led some very large studies of the NF1 mutation patient groups. He's also the director of the Heflin Center for Genomics, Genomic Sciences, and he's really worked strongly together with Hudson Alpha to set up an additional genomics and genetic research cooperative. That's so he's a co-director of the Hudson Alpha UAB Genomics and Genetics Center, I guess. His trainings did all of his training in Boston at Boston Children's Hospital and Harvard Medical School, and we're really lucky to have him here at UAB. So thanks for me.
Great, thank you, David. So I was asked to review some of the resources in research that would support personalized or precision medicine, whatever term you like to use. This word, but it does. So, one question to start with is, what do we mean by personalized or precision medicine? And I think it really boils down to what do we actually mean by the concept of individuality. And you can define that in terms of a number of components. This is not meant to be an exhaustive list, but ancestry really is basically genetics, what you have inherited, dating back any number of generations. Of course, individuals will have over time, unique environment that they've grown up in. There are cultural differences in terms of how people, let's say, respond to conditions of health and disease. And then a person's experience, how they view their own health may all contribute to individuality. And Where this is converging is towards the ability to define risk of disease, the way an individual might respond to treatment, and what sorts of side effects of treatment they might be vulnerable to. So when we talk about personalized medicine, I see it as an integration of all these things, and I'll focus mostly on genomic medicine because that's the research domain that I'm most familiar with, but I wouldn't want you to come away thinking that genomic medicine and personalized medicine are synonymous. Genomic medicine is one exceedingly powerful way to individualize care, but it isn't by any means the entire picture. So I'm going to approach this by looking at a few different domains. We're going to begin with rare disease, which is the area where genomics has probably moved the fastest in terms of adding insights. Common disease, where there's huge need for better understanding of pathogenesis. We'll talk about cancer, which has also moved pretty far in terms of individualization of management. Epigenetics, because it's an area where there's been a lot of interesting discussion about what could be going on and where that contributes. And then I want to end by talking about the collaboration that David mentioned a few minutes ago with Hudson Alpha, but actually beyond that also some initiatives that are happening here at UAB. So I'm going to start with something that might seem a little bit off topic, but tells an interesting story and gives you an idea of what the power of a genomic approach to a rare disease can be. So it's probable that most, if not all of you, have never seen somebody with progeria, but I'll bet you've heard of it just because it's such a dramatic phenotype of children who undergo very rapid premature aging, in fact die usually in their teens of the various complications we'd normally associate with old age. It's vanishingly rare, just probably a few hundred patients at most ever identified with this, and it's always sporadic. So it always happens in just an individual in the family with no family history. So what would be the reason to think it was a genetically determined disorder? Now you can imagine it wouldn't be passed from generation to generation because people don't usually live long enough to reproduce. But the exceedingly stereotyped phenotype from one person to the next, I think, was the strongest argument in favor of a genetic basis. And to make a long story short, the gene, in fact, was identified. It's lamin A, and the particular mutation is always the same in everybody who's affected, which is substituting a T for a C in exon 11. And what that does is cause a new exon splice donor to be generated. So normally a splice between exons 11 and 12 would occur at the end of exon 11 and beginning of exon 12. But here, a new splice donor is recognized internal to the exon, and when that, which by the way competes effectively against the normal one, and so what it does is it removes some bases from the end of exon 11. And why that matters is lamin is a nuclear membrane protein. It lines the inner nuclear membrane, and as it's formed, it actually binds to the membrane by a reaction called farnesylation, where it binds an isoprenoid group that anchors it to the membrane. And then as the cell matures, a cleavage site is recognized by an enzyme, and it cuts there, releasing the lamin where it then coats the chromosomes. If you're missing the enzyme, this is a mouse model, that won't happen. But if you have progeria, you're missing the recognition site for the enzyme. And as a consequence, the lamin never gets released from the inner nuclear membrane. It's permanently anchored there, and it fails to attach to the chromosomes that suffer over time DNA damage. You get these nuclear blebs over time. And this seems to be ultimately what is reducing the lifespan of the cells, which in turn is what's causing the accelerated aging. Why I mention this is, for 100 years, whatever, the phenotype was recognized and nobody had any idea what was going on. When the gene was discovered, it revealed an underlying pathophysiology And that in turn actually has spawned clinical trials because it is possible to inhibit the reaction that binds the lamin to the nuclear membrane and effectively release it. And now there have been clinical trials with drugs that have shown some effectiveness. That would, I 100% guarantee, not have happened. Nobody would have ever had a clue that those drugs were relevant if it weren't for the fact that the gene was identified. So there is a lot of value to finding the gene, aside from the genetic counseling implications that it may convey. So let me walk you through quickly some of the technologies that have evolved in these past few years to get at the genetics first of rare disease, and then we'll switch gears to common disease. So you're probably familiar with the sort of karyotype, which is a picture I actually pulled out of a lab file from the 1960s. It shows 47 chromosomes. You'll recognize the phenotype of trisomy 21, which was the first example of a chromosomal trisomy syndrome identified actually in 1959. So that's what chromosomes looked like around that time. This is what they look like in the laboratory today, using microarray technology. It's done in various ways. One is to combine DNA from a patient with a reference DNA, color one, say, in red, one in green, let them mix, and then compete for hybridization on a chip containing oligonucleotides from all over the genome. And each dot represents a hybridization signal, and if they are about equal, then they lie on this kind of midline, but you may notice way down here a shift to the left, and this represents a deletion that was found in the patient's DNA. So there was excess hybridization of the control, which in this case was labeled green. You may or may not be able to see the green from where you sit. And it diagnoses a deletion that can be clinically significant, but would never have been visible, certainly not with the picture I showed you a moment ago, not even with more modern cytogenetic analysis. Whereas the microarray analysis now is detecting copy number changes, deletions or duplications in upwards of 20% of individuals with unexplained intellectual disability or autism spectrum disorder, hence this picture. So it's really completely changed our approach, and now is the first line test when dealing with those kinds of phenotypes. Meanwhile, the ability to recognize the genes that underlie various genetic disorders has also gone through a huge sea change in recent years. So this is a kind of diagram of how the cystic fibrosis gene was identified, which was in 1987, which probably sounds like a way long time ago. So it was first identified through family studies that the gene resides on a site on chromosome 7. And then a positional cloning exercise was done, more complicated by far than I'm depicting here, but having genetic linkage with DNA markers that were near allowed one to kind of walk along the chromosome, actually really jump from marker to marker, each time converging on the gene itself, and finally identifying a gene that was consistently mutated in individuals with cystic fibrosis and which was expressed in lung, and you can actually then develop genetic tests as was done. This was discovered to be a chloride channel, and it has actually opened the door to therapeutics, and in fact I'll say a little more about that. So the idea here, this was one of the earlier examples of a gene that was identified by its location in the genome, not with any prior knowledge of the actual function of the gene product. And it's actually a little bit sobering because it makes you wonder kind of, you know, what am I doing about which people will say the same thing. But the sobering thing is that you could take all the literature on cystic fibrosis, at least the genetic literature, prior to 1987 and lock it in a library and throw away the key, and the world would be none the less for it, because really none of it turned out to be relevant to the etiology of CF until the gene was discovered. And this gene was on nobody's list because no one knew it existed as a candidate for CF. the candidate gene approach, where you have an enzyme and you say, well, that must be the culprit in this particular disorder, that got played out, in the late 70s and early 80s, and now it really became necessary to use other tools. So this has really taken off exponentially, as you can see, in terms of the pace of gene discovery, and it isn't over yet, by the way. Of what are estimated to be about 7,000 Mendelian traits, we still have knowledge of the genes only for a proportion of those. So what's really changed the approach is the ability to do what is often referred to as next generation sequencing. This is a data set that is maintained by the National Human Genome Research Institute, and it's looking at the cost of sequencing over time, sequencing a genome this is. And notice the y-axis is an exponential axis. So in 2001 it cost $100 million to sequence a genome, which puts it out of the range of most individuals. And in a time frame from there to about 2007, the price came down tenfold to just $10 million, which is a pretty impressive price reduction, but still out of the range, certainly, of what you could expect to use clinically. That price drop more or less tracked with Moore's Law. You may be familiar. This is a statement of the ability to increasingly pack transistors onto a computer chip. And it's how come the computer that probably all of you have in your pocket is more powerful than the one that would have filled a room this size in the 1970s. And there was a kind of neat thing that sequencing was tracking Moore's Law, doubling about every 18 months in terms of, in this case, cost reduction. But then you'll notice in 2007 it really started plummeting and far exceeded Moore's Law. Barely can see it off to the corner here. But in 2015 it dropped again, and it's kind of hovering very close to the $1,000 range, which that's a pretty impressive price reduction from $100 million just 15 or so years ago. And it's nice to see that it's really exceeded Moore's Law until you remember that you need computers to actually analyze the genomic information. So it's not always such a wonderful thing. But nonetheless, now the ability to do genome sequencing is practically routine and happens on machines that sit on a desktop. I won't try to walk you through in great detail how next generation sequencing is done because the chemistry is constantly changing and there's a lot of different approaches. But just to give you a kind of very quick hand-waving appreciation, just imagine a solid substrate like a glass chip, say, onto which you adhere chunks of DNA, just 100 base pairs in length, and you amplify them, so where there was a single strand, now there are multiple strands. And you use them as a template for a DNA synthesis reaction, so where there's an A T will be incorporated, and where there's a G, a C will be incorporated, and so on. And you color code the 4 bases, in this case, red, yellow, blue, and green for the A, T, G, and C. And then you allow the reaction to go 1 base at a time. So you let the first base find its homolog on the opposite strand. You freeze the reaction for just a moment. And now picture, if you will, that the chip is like a tabletop and that there's a camera in the ceiling looking down on it. And it snaps a picture after each base binds. So the first time, this strand here bound a red thing, that's an A, and the next door strand bound a green thing, so that's a C, and so on. You allow the next base to bind. This one now has a blue, which is a G, as does this one, it's also a G. Anyway, you let that happen. It only has to happen 100 times, and you've sequenced the whole genome. Now, it's true you've only sequenced it in 100 base pair fragments, and there are 3 billion base pairs in a haploid genome. But since we know the reference sequence now, a computer can take these 100 base reads and match them to where they belong in the sequence of the whole genome. And in a matter of what could be just hours now, you can be sequencing the entire genome where it would have taken, well, honestly, years in the past. So it's often referred to as next generation sequencing or massively parallel, because you're doing parallel sequencing of so many different strands. Now, this is in constant evolution. It's a grossly oversimplified view of it, and there are a lot of different chemistries, so this is not the only way it's done. But why I show this is it gives you a kind of visual image of what's really going on, and well, obviously it's not magic. So here's the first published case that was solved using this. It's an exceedingly rare disorder called Miller syndrome. There had only been a handful of people ever described with it. You can see facial dysmorphism, abnormal limbs, and intellectual disability. And what was done was sequencing of actually the expressed part of the genome, the exome, which I'll come back to in a minute and define a little more carefully. And they did it on 4 individuals. So they were actually 3 kindreds, 3 families. One had 2 affected SIVs. That's kindred 1A and 1B. And then there were kindreds 2 and 3. Okay, so 4 individuals got their exome sequenced. And what they did then, just focus for a minute on this here, was they applied some filters. They said, we're only interested in things like nonsense mutations and insertions and deletions, things that would be significant alterations of sequence. They found just in individual 1A, 4,670 of these that are dominant, meaning only one allele was affected, and 2,863 that were recessive, meaning both alleles had a variant. Now they said, well, this is a really rare disorder, and if it's that rare, it's not going to be found in any database of common variants. So in this case, they took a couple of databases of common variants and filtered, and for example, of the 4,670 single allele dominant variants, only 456, so about 10%, were not found in either of these catalogs of common variants, and only 31 were affecting both alleles. Then they did a computer algorithm and they said, well, can we model these and identify variants that are probably damaging to the protein, because this is obviously a significant phenotype, and of the 456, only 204 that were dominant and only 6 that affected both alleles. That's person 1. Then they looked at the same thing with person 2, found similar numbers, and then they asked, on these 2 lists, is there anything in common? It turned out there were 83 things that they shared that were predicted damaging, rare, and would have significant mutations that were single alleles, but only 1 that affected both alleles. They then looked at kindred 2 and 3 and compared them to this, and this same 1 was also mutated in both of these other unrelated individuals, and that was the gene. So this is finding a gene based on looking at just 4 people, and it was such a rare disorder, there were no other people pretty much to look at. You couldn't do genetic linkage because there were no large families, but they zeroed in on the gene nevertheless. So they did exome sequencing. The idea is you take excuse me, genomic DNA, allow it to be cut into fragments, and then you hybridize this DNA against a set of oligonucleotides that have been synthesized that correspond to all of the exons, all the protein encoding sequences in the genome. You then wash away everything that doesn't bind, which is all the DNA that's not encoding protein. You sequence it and align it. So what you're looking at then is about 1.5% of the genome, which is the only amount that's actually encoding protein, which is a kind of daunting thing to remember, that everything you learned about genetics really only applied to 1.5% of the genome. But that's the way it is. But it also turns out to be where most, maybe not all, of the mutations that cause disease seem to reside. So this is showing a list of how many variants you're likely to see in individuals where you would subject them to exome sequencing. Hundreds of missense, a few nonsense, synonymous meaning they don't change the amino acid, but thousands of variants that have been seen before, several that are not. It's interesting to notice that the number of variants tends to be greater in, this is in the American population, in individuals of African American ancestry rather than European. And the reason for that is that because populations originated in Africa, there were fewer bottlenecks in migration. So European Americans derived from a small subset of originally Africans who eventually migrated to places in Europe. And so their genetic diversity was less because they were derived from a handful, probably more than a handful, but a smaller number of individuals, whereas African populations are the ancestral populations of humans. So you tend to find more variants in Africans or people of African ancestry than of other ancestries. In general, how do you do this? If you work on a rare Mendelian trait, there are a few ways to do it. What I just described is looking at multiple different individuals from different families and looking at the intersection of what do they have in common. Figuring if it's the same disorder, then they probably share a gene which is mutated, whereas other genes that will be variant are not likely to be the same. So that's one way to do it. Second way to do it is would have worked in progeria in retrospect if it had been available at the time, and that is you look for things that are new mutations. I didn't say this before, but the reason progeria is so rare is that it can't be passed on, right, because people who have it don't reproduce, and therefore every case is a new mutation. And the reason it's vanishingly rare is the mutation has to be a surgical hit on just that one nucleotide in the lamin A gene. So it's going to be a rare chance that it happens. But if you are dealing with new mutations, you can sequence the child and both parents and ask, what does the child have that the parents don't? And that doesn't diagnose it, as you'll see in a minute, but it can help a lot. Or you can look at big families, if you happen to have them, and sequence multiple individuals and ask what they have in common. Or you can take lots of individuals with a phenotype and compare outliers, people with no phenotype and those with severe phenotypes, and ask what's different between them. So we're doing this all the time now. We have the ability to do genome sequencing in the Heflin Center here on campus. We also collaborate closely with Hudson Alpha. They do whole genome sequencing. They don't bother selecting the exons because the machinery they have available can do genomes faster actually than exomes. And you might get a few 100,000 variants on an exome and 4 or 5 million variants on a genome. But nonetheless, it is a ultimately more powerful approach if you can go to the whole genome. Here's a case in our undiagnosed diseases program that we solved. This is the child here with a seizure disorder and brain abnormalities, very small pons and very large cystic area by the cerebellum, severe intellectual disability. Interestingly, he had 2 variants, both of which were de novo. SP10, there was a variant, and you notice both parents are wild type, not showing the variant. And SETD5 was also heterozygous in the child, but homozygous wild type in the parents. Turns out, this is a published case also showing hypoplasia of the brainstem and the cerebellum, that the SP10 gene is probably the driver, if you will, for this phenotype. The other gene, we're not sure what it's contributing. It's associated with intellectual disability. But this turned out, after we found it, to be an already described variant. We had good evidence that it was pathogenic, and it almost surely is the etiology of this child's problems. It's de novo, meaning it's a new mutation, and in fact this child had 2 new mutations. And that might bother you. You know, we're kind of brought up on the Occam's razor theory, you know, the idea that you choose the simplest explanation, and in a medical disorder, the simplest explanation is a person only has one problem. And that's turning out not to be the case in a lot of instances. If you sequence a child, just anybody, and you sequence both parents, you'll find in round numbers about 60 new mutations in every child. Every one of us has 60 roughly new mutations compared to our parents. Okay. That's actually an interesting and somewhat daunting figure, because you can calculate a mutation rate on a per nucleotide basis. It's in the 10 to the minus 8th per nucleotide per generation. We have 3 times 10 to the 9th, so that's why you get about 30 mutations in every sperm cell and every egg cell. And if you step back and think about it, if you consider the number of sperm and egg cells there are, what it's telling you is that every one of us in our germline carries every gene mutation that's ever been described or ever will be described. And it maybe would be a kind of a daunting thing in terms of reproduction. How does it ever work if there are that many mutations? And of course the answer is there's a lot of space in the genome, and most of these mutations reside in between genes or in places that are not critical to gene function. So it's a little bit like a meteor falling into the middle of the Pacific Ocean in the middle of the night, not near an island or a ship. You'd never know what happened. But once in a while, rarely, but once in a while, it actually hits something on land that makes a difference, and that's what you notice as a genetic disorder. Most of these are silent because they don't affect gene function. Not going to go into detail here. I just wanted you to realize that saying that a variant is pathogenic is a lot trickier than you might imagine. When I said a minute ago that this variant in this child had been seen before in a published case and caused a very similar phenotype, that gave us reasonable confidence that we were dealing with a true pathogenic variant. But very often it's not so straightforward. We find private variants never seen before, never described before. They look compelling because they might be de novo, or they might be in a gene which is known to be physiologically related to the condition. But how do you prove that a variant never seen before is pathogenic?
Can I ask a question about on the slide just before this?
Sure.
So if people have, in general, 60 mutations, and using your analogy of the meteor falling in the ocean and not hitting a ship or land, could that mutation at some point further down hit land?
The actual mutation itself? Well, you know,
is that a characteristic of the mutation?
it's a characteristic, you know, that particular mutation, if it landed in an area that wasn't really functional at all, it's doubtful it would ever have an impact. I guess the one circumstance it could is if it's a recessive. So you have a single nucleotide change now, but it would have required both alleles to be mutated to actually get a disorder, and so it could have an impact, you know, generations later. By chance, a partner happened to carry a mutation in the same gene. So it might be silent because it's recessive and surface someday generations later as a homozygous individual. But, you know, for the most part, I'm sure there are nuances to this. You know, there might be effects of having multiple mutations together that no one alone is enough to cause a phenotype, but together they do. But for practical purposes, most of these are silent mutations.
Thanks.
Yeah. So, you know, again, there are pretty now clearly defined standards for how you decide that something's pathogenic. And I'm not going to walk you through this. It'll be tedious and complicated. I forgot to put the reference on here, but the American College of Medical Genetics and Genomics and the College of American Pathologists got together and made these standards where they define levels of evidence for pathogenicity or for something being benign. And there's, you know, either very strong to supporting evidence or the same for a variant being benign. And you can't read this, but this is just pointing out the fact that there are some very clearly defined criteria. An example would be a de novo, meaning you sequence both parents and they don't carry this variant, but the child does. That tends to be strong evidence of being benign, but not proof, being pathogenic, but not proof, because I just said you can have things that are de novo that don't reside in genes. It needs to be proved that that's the case. You have to actually show not only that both parents don't carry it, but that they really are the parents. So you actually do identity testing to confirm that yes, they are the parents, but no, they don't carry it. One of the strongest pieces of evidence is that there are other cases described with the same phenotype and the same variant, which was the case in the patient I described a few minutes ago. But there's all different levels of evidence that on the one side, the spectrum is from pathogenic to likely pathogenic on the extreme end, or benign or likely benign. We don't tend to pay much attention to those. Right in the middle is something called variant of unknown significance, which means that it's variant of unknown significance. You don't want to use it for clinical decision making, and I've seen lots of errors made where people did. They saw this variant, it looked really tantalizing, but if it's labeled a VUS, don't use it for clinical decision making, because it could easily turn out, as more evidence accumulates, that it'll be proved to be benign. One of the things nowadays that we can do is compare outcomes in an individual being sequenced with basically the rest of the world. This is a website that has tried to federate databases. What's often used is Gene Matcher, and basically you put in to this database what you found phenotypically and at the genomic level, and it goes out into a database where anybody else who's ever seen the same thing before and put it in will constitute a match. And there are many examples where exceedingly rare genomic changes were identified and then verified through Gene Matcher. We do use a very standardized, what's called human phenotype ontology to describe phenotypes, so it's not just, kind of casual terms, and there are websites that you can use to look at that. It can make a big difference. I told you about progeria and how finding the gene spawned the possibility of treatment. Tuberous sclerosis complex, a single gene disorder, turns out to be a mutation in either of two genes that regulate a protein called mTOR, and a drug now inhibits mTOR and actually has been successful in shrinking the various tumors associated with tuberous sclerosis. Or in the case of cystic fibrosis, we talked about before, there are now a couple of drugs that are FDA approved. This is showing one that actually restores function to the mutated gene product. And here's a clinical trial showing pulmonary function improved in those treated compared to placebo. So knowing the gene mutation can actually open the door to devising specific targeted therapeutics. So that's for rare disease. Let me switch gears and talk a little bit about common disease, multifactorial conditions like asthma, hypertension, and diabetes, where no one gene is responsible for all cases. The kind of paradigm is that if we are all born with some genetic liability to common disease, but unlike progeria, it's not deterministic. It doesn't guarantee you'll get the condition. But over time, and maybe the exposure to environmental factors, you cross the kind of threshold of disease. And the question is, if we knew what this genetic liability was, could we identify these individuals, maybe help avoid these exposures, or if you do get disease, understand better how it works and devise better treatments. It's been really challenging, though, finding the factors that predispose to common disease. One that has been in play for about the last 15 years is looking at association studies, taking two alleles that differ in a single nucleotide, the so-called single nucleotide polymorphism, and asking, is there an association to a common disorder? You can do a case control study. 1,000 people with asthma, 1,000 who don't have asthma, and you find allele 2 in 30% of those with asthma, but only 10% of those who don't have it, which, with appropriate controls, could confirm the hypothesis that allele 2 is associated with asthma. Doesn't mean it causes it, could, but it might be that it just is linked to another gene that you haven't identified, which is really contributing, and this one is just a surrogate marker. One of the first successes was age-related macular degeneration, where a couple of alleles were found in single nucleotide polymorphisms searched across the entire genome. This one turned out to be complement factor H. Nobody had any idea it was involved in this disorder until an association study indicated that it was. And this is a diagram now maintained by a group in the UK. Each color represents a different common disorder, and each is pointing to a place on the chromosome where an association has been found, huge number of associations. not necessarily strong associations in every case. Sometimes they are, sometimes they're modest associations. And as you'll see in a few minutes, not necessarily so useful in clinical practice, but potentially very useful in terms of understanding pathophysiology. But you need to realize that with all this effort, there's been a lot, as you can infer from that previous slide, if you take estimates of the genetic contribution to a common disorder, which is referred to as heritability, what you may find, and this is a theoretical case where heritability is calculated to be 50%. So that means 50% of the variance in this phenotype is accounted for by genetic factors. What you might find is a small proportion, in this hypothetical case, 15% has been accounted for by genome-wide association studies, meaning 30% is so-called missing, meaning no one's found the factors. And it may be because a lot of the factors you can look at with GWAS studies wind up being common variants, and maybe many of the factors are not so common variants. Or maybe the heritability has been overestimated, or maybe it's not single nucleotide polymorphisms, but some other, such as copy number variants, that account for this. But you do need to realize that we've been chipping away at the edges of the contributions to common disorders rather than any one case where we can explain all of it. It has spawned interest in including direct-to-consumer testing. This is an old screenshot when I did 23andMe. Some of you may have done it or heard about it. They don't return this kind of information anymore because the FDA stopped them from doing it. But it showed me to be at average risk of type 2 diabetes through this mechanism. I knew enough to take that with a grain of salt, meaning it didn't render me immune to type 2 diabetes, although there's concern some people might conclude that. And a few years ago, when there were a few companies that were doing this kind of SNP analysis, 5 people sent their samples to 2 labs, and when the arrows point the same way, they got essentially the same outcome. But here are several examples where they contradicted each other. And it turned out, when you look behind the scenes, they almost never disagreed about the genotype. What they did disagree on is how they calculated risk. And that, I think, is one of the things that led to a skepticism about using these outcomes in predicting risk. But that's not to say they're unuseful in terms of understanding pathophysiology.
Is that because of the role of the environment?
Part of it is differences in environment, but part of it is just different assessments of the contribution of particular markers to disease. And also, you know, some of these studies are done in particular ethnicities that may not apply to backgrounds of other individuals. Well, very quickly, let me just say a few words about cancer, which is a multi-step process starting from a normal cell acquiring genetic change that eventually leads to malignancy. You now can sequence a cancer, if you wish to, compare it to normal tissue, and catalog all the genetic changes, and in many cases use this as a basis for defining therapeutics. Here's an example where the HER2 new receptor is amplified and a particular antibody can be used to treat if that's present. Here's a particular BRAF mutation which, when present, responds to a specific chemotherapy. This is being done routinely. If you want to hear more about it, today at 3 we're going to have a seminar in the Genomic Medicine series on doing this in the context of ovarian cancer. We all have some circulating DNA in our blood from cells that degenerate and release their DNA. That includes tumors, and now there's a lot of interest in actually sequencing this DNA in your circulation to have it be a sort of early kind of warning sign of the presence of cancer. By the way, it's used all the time now in prenatal diagnosis because 10% of the circulating DNA during pregnancy is derived from the fetus, and so you can actually infer information about the fetal genome by doing a blood sample from the mother. Very quickly, let me say a few words about epigenetics, defined originally long before the era of DNA technology as a heritable change in gene expression, but not associated with a change of base sequence. And we don't mean heritable from generation to generation of an organism, we mean from cell to cell. And it usually is explained in terms of changes at the promoter region, including methylation of the promoter and also modifications of proteins, especially histones, that either would lead a piece of DNA to be compacted and inaccessible to transcription, or relaxed and open and accessible if the promoter is non-methylated. You can have DNA methylation in the promoter, as I just described. It can also be present inside of a gene. And the methylation in question is mainly 5-methylcytosine, typically occurring in the promoter at so-called CPG islands, cytosines followed by guanines. And if you look at the way that behaves in cell replication, which is why it is thought of as a heritable change, the methylated cytosines are shown in red, the guanines nearby in yellow. There is no pool of 5-methylcytosine to be incorporated into the DNA. So when the DNA replicates, normal cytosine, shown in blue, is incorporated adjacent to each guanine. So you might say you're going to lose your methylation signal, but there are enzymes that recognize the one strand is methylated, and when they see a CPG, as they do here, one of which is methylated, it methylates the other one. So it's the enzyme system that maintains the methylation pattern for a particular gene from cell generation to cell generation, although all of these methylation marks tend to be erased in germ cell development. I'm not going to go into detail here except to point out there are lots of modifications to the proteins, especially the histones, the major proteins that comprise the nucleosome, which is the major unit of organization of chromatin. So this is not purely a DNA modification. And there are many proteins that recognize the methylated promoters with modified histones and prevent transcription from occurring and compact them. So it's a complex process. An important point to realize about the methylation of DNA is that it is tissue specific. Some people say, well, so can we characterize the epigenome of a given individual, hypothesizing maybe certain genes are turned off by methylation, certain are turned on, and they'll say, well, we'll do a study where we'll draw some blood and we'll assess methylation, which definitely can be done, and infer that. And the point to realize, what you're looking at is a heat map. Yellow is no methylation, blue is high degree of methylation, for 4 regions of a chromosome in lots of different tissues. And the point is that you see differences in methylation of any of these regions in different tissues. So there is no single epigenome. There's as many epigenomes as there are cell types in an individual. And if you're going to study epigenetic changes, you need to study a tissue that's relevant to the phenotype that you're interested in. All right, 2 more things in terms of tools that I'll talk about, and then I'm just going to end with some resources here, and I try to do this quickly. One of the main drivers in personalized medicine is the idea of pharmacogenetics, alterations of how drugs are metabolized due to genetic polymorphisms. This can affect absorption, metabolism, and excretion. There are phase 1 reactions that usually are oxidation reactions on drugs or chemicals of any kind, and phase 2 reactions are conjugations that usually render things soluble for excretion. And there actually is a phase 3 which has to do with transport in and out of the cell. There are lots of genes that control these processes and have polymorphisms that determine that is either rapid or slow enzymatic activity. We tend to describe these in terms of what are called diplotypes, a term you don't usually hear in genetics too much. If you consider 2 gene loci, A and B, that are on the same chromosome, and then each has 2 alleles, A1, A2, B1, B2, we call A1 and B1 a haplotype. They're on the same chromosome together. A2 and B2, likewise a haplotype. You could say A1, A2 is a genotype, as is B1, B2. But you can also say having the genotype A1, A2 and B1, B2 and the haplotype A1, B1, A2, B2 all together constitutes a diplotype. And it's the complexity of the combination of alleles at multiple loci that define many pharmacogenetic polymorphisms. And rather than describe them in terms of A and B, it's convenient to say an individual with A1A2, B1B2 is a diplotype that usually is labeled with a star and then a number for what particular diplotype it is. So very quickly, a few key examples. The various cytochrome oxidases involved in phase 1 reactions. CYP2D6, lots of drugs, and this is a very short list of all the drugs are metabolized. And if you're a slow metabolizer and it's a reaction that would normally inactivate the drug, you're going to accumulate it and potentially have side effects. Or if you're a rapid metabolizer, the opposite. A couple of polymorphisms of importance. CYP2C9 is involved in warfarin metabolism and excretion. Individuals who are slow metabolizers will accumulate more warfarin and be vulnerable to hemorrhage if they're treated. And there's also a polymorphism in the protein target of warfarin, VCORC1, which, if that is altered, it might change sensitivity to warfarin. So you can use these two actually to predict the ideal dosage for an individual, avoiding either under medication and blood clots or over medication and hemorrhage. Clopidogrel, an antiplatelet drug, is really pharmacologically inactive until CYP2C19 activates it, and if you don't have activity of this, clopidogrel will be useless. And there's a FDA recommendation that testing be done to predict individuals who are going to respond versus those who are not. Thiopurines methyltransferase is involved in inactivation of drugs used in treating leukemia and autoimmune disease. Small proportion of people have low activity, which means they accumulate this drug, and these are the people who will develop potentially fatal bone marrow failure treated with a standard dose, and because of the devastating nature of that reaction, it does often get tested. An acetyltransferase accounts for some toxicity due to treatment with things like isoniazid and hydralazine. A whole slew of drug transporters, there's not really time to go into them. One you may have heard of is associated with statin-induced myopathy, and you can predict who's likely to get that based on testing. This is an interesting story. There's a particular HLA polymorphism found in the Asian population, mainly in China and Southeast Asia, that predisposes to Stevens-Johnson syndrome, toxic epidermal necrolysis. After treatment with drugs like carbamazepine, it's common enough in those populations that in some areas everybody gets tested and they carry a card and it says, don't treat me with carbamazepine or other drugs that are at risk here if I ever should need to be treated. That doesn't work outside of that region because this particular HLA diplotype is not as common outside of Asia. And the ones that are associated are really not, no one of them is common enough to make it possible to do universal testing. There is a pharmacogenetics knowledge base, it's a website, and then there's a consortium called CPIC, you can see what it stands for, that produce peer-reviewed guidelines. So in the last couple of minutes, and I guess I do mean a couple of minutes, let me just make a couple of points about resources here on campus. There is a genomics core laboratory in the Heflin Center, does all the things we've been talking about. I didn't mention RNA-seq and ChIP-seq, and there is bioinformatics support. RNA-seq is just a way of making a library of RNA from any cell type or tissue and sequencing it and characterizing the full range of RNAs that are expressed in that tissue. We don't do RNA microarrays anymore. This is a much more sensitive and quantitative approach. ChIP-seq amounts to taking antibodies to particular proteins that might bind the chromatin. You use them to immunoprecipitate the chromatin, then you wash away the protein and sequence it, and it's a way of figuring out for a given protein that binds to DNA, what sequence does it recognize. This is showing an example of network analysis. David Crossman in the Heflin Center can help you with bioinformatic analysis. And then David mentioned the UAB Hudson Alpha Center, which Hudson Alpha, if you haven't been there, is in Huntsville, about an hour and a half north. We had a symposium there last week, and we actually do that once a year. Actually have a bus that takes people up there if you don't want to drive it. And A lot of different research collaborations have been spawned. We do an immersion course. I don't know if any of you have participated in that. In the fall each year, this was the syllabus last year. It's Monday through Friday, 8:30 to 12:30. A set of lectures about application of genomics, many of the things I've talked about today, but amplified quite substantially. Usually about 24 people take it. And then in the spring, which you'll notice the dates happens to be this week, actually as we speak. This is happening. David Crossman actually walks people through data analysis using real data sets in real time. It's happening over in the Finley Center this week. And that's, I think there were 30 people that signed up for that this time around. Let me advertise that we have a T32 funded by NHGRI. In genomic medicine, we have an open slot right now, and we're looking for MDs, PhDs, or MD PhDs, or any doctoral degree actually, interested in spending 2 years training in genomic medicine, and we would welcome applications if any of you know people that would be interested in doing this. We have a seminar series on genomic medicine, which actually will happen today at 3 o'clock, focusing today on cancer genomics. We have a lecture series on bioethics in genomic medicine, which happens once a month, alternately at Hudson Alpha or here, but we stream them across either way. We just announced an RFA for systems-oriented initiatives in different sort of medical systems. We'll be offering $75,000 to $100,000 a year times 2 years for individuals interested in proposing systems-oriented collaborative genomic initiatives between UAB and Hudson Alpha. And finally, let me mention that actually it officially launches tomorrow. We have money from the state, $2 million this year, to launch the Alabama Genomic Health Initiative. We'll be offering genotyping or sequencing to 10,000 people in the state, of whom most will be healthy, or at least not selected for any particular phenotype individuals, and they'll get a return of results of actionable genes, of which right now the list is 59, and then a risk assessment, a risk mitigation plan. For example, a breast cancer predisposing gene might be discovered, and then we'll provide them genetic counseling and an approach to dealing with that risk. We'll also be doing whole genome sequencing on a subset of individuals. And anybody wants to get involved in this, let us know. There'll be research opportunities. We're creating a database and a biobank along with this. It'd be a perfect sort of project for somebody involved in the T32. The UAB Hudson Alpha Center has a newsletter, and here are the two coordinators, Ida Starling at Hudson Alpha and Shaila Handatu here at UAB, and their contact information is listed, and they can easily get you a subscription to the newsletter. All right, we're only 1 minute over. That could have been worse. And if there are questions and there's time, I'll be glad. Yeah.
What's the ethics surrounding, especially the latter collaborative you discussed, the Alabama genetic? So for instance, if you incorporate 20, 30-year-olds who now have known variants or perhaps pathogenic variants, is that disclosable? Do they need to disclose that for insurance purposes? Those sorts of things.
Yeah, so, first of all, we're very aware of the ethical implications. We have a ethics working group, and, this has gone through the IRB, and we have full IRB approval for this project. And we're just now working on a certificate of confidentiality, which means that the data can't be even subpoenaed. With all that said, there is a law, a federal law called the Genetic Information Non-Discrimination Act, which prevents the use of any genetic information by employers to make employment decisions or health insurers to use it as a basis either for issuing insurance or for determining insurance rates. You're not protected, though, for life insurance and disability insurance. And that's a point that is mentioned in the consent form. And I guess you can, if that bothers you as a participant, then don't participate is, the obvious answer. I guess one way of looking at it is, would you rather have life insurance or let me say this differently, would you rather take the risk of losing your life insurance, or would you like to have to use your life insurance, which is in effect what it's saying, because the 59 genes we're talking about, you know, one of them, or one, not just one group of them predisposed to arrhythmia and sudden death. So if you're so concerned about your life insurability that you don't want to be tested, and it turns out you're in the 1 to 3% of people that have such a variant, not only cardiac, but cancer and so forth, you know, then you're at risk of sudden death, and the fact that you were able to maintain your life insurance is probably going to be scant consolation. So I take the point of view that it's a decision everybody has to make for themselves, but the value proposition from our side is that it's possible it will interfere with your ability to get life insurance, but it also can save your life. And that's the perspective we're taking. Yeah.
When it comes to the epigenome inside of the same tissue, is it normally consistent or not necessarily?
Actually, it really would be a function of cell type within a tissue, probably. It probably would be pretty consistent with a given cell type, but the farther, you know, skew you get from a specific developmental pathway, the particular genes, when you think about it, you know, why is a fibroblast different from an epithelial cell? It's basically through turning off certain genes and turning on others. Many of the genes that are modulated that way are modulated through epigenetic changes. So you would predict, if you took a skin sample that had both epithelial cells and fibroblasts, you'd expect different epigenomes in those two cell lineages. Okay. Well, thank you.
Thank you.
Yeah.
A year from now. The general immersion course will happen, I guess, sometime in October, probably. And usually the sort of immersion course happens in May.
We can ask somebody if let me know.
No,
What will we?
the 21st.
We're trying to keep it a day so that everybody can have time to do things.
You too.
Okay. So should I just try to?
I can send you the list
Okay.
and let you know, and then that'll give you people different times and stuff, and see if they don't want to switch.
Okay.
Because you've got this problem that's in that.
Okay. That'd be great.
And then if you try to get people, and there's just no way.
Yeah.
Sneak in.
Okay, you have a good one.
You too. So, what did you learn about, actually, turn this so that we're not taped.